Hi, so I'm Niamh. I've just started the final year of my PhD at the University of Warwick, developing an ex vivo pig lung model of Pseudomonas aeruginosa biofilm infection of the cystic fibrosis lung. So why are we developing a new model for these biofilm infections? These can broadly be divided into four key aspects. The first being the genetic condition we're focused on, cystic fibrosis. One of the key aspects of this condition is defective mucociliary clearance, which results in an ideal environment for bacterial infection. These infections tend to follow a timeline as shown on the screen, when initially you see Staph aureus infection dominating before people age sort of into their teen years, where you start to see intermittent Pseudomonas aeruginosa infection. As these infections go on and people enter their 20s and 30s, you start to see these chronic Pseudomonas aeruginosa biofilm infections, as well as infection with other gram-negative species and fungal infections. These infections play a key role in leading to the progressive respiratory failure often seen, which tend to require lung transplantation. Although we know a lot about these infections, it is still unknown what drives this change from intermittent infection to chronic biofilm infection and what makes these infections so difficult to treat. I'm specifically focused on the biofilm infections, so the chronic established infection. This is when the community of bacteria live within a self-produced matrix made up of extracellular DNA and extracellular polysaccharides. These act as a protective layer for the bacteria against the host immune response and antimicrobials. Also, these bacteria do not escape the rise in incidence of antimicrobial resistance and all the traditional mechanisms you would expect to see. But even if you develop an antimicrobial antibiotic treatment that these bacteria are susceptible to, often they can't penetrate the biofilm to get to the bacterial cells, so they can't eradicate them. So I'm specifically focused on those biofilms caused by the dominant pathogen Pseudomonas aeruginosa. It's thought that around 75% of adults with cystic fibrosis in the UK will become infected with this bacteria during their lifetime, whether this is just the intermittent acute infection or this established chronic biofilm infection. Although it is one of the most well-studied pathogens in one of the most well-studied biofilm infection environments, we still don't understand what drives this chronic infection and the periods of acute exacerbation experienced where the bacteria revert to a highly virulent lifestyle. Because it is so well studied and we already know so many aspects, it also makes it the ideal infection to focus on when developing a novel model to try and get closer to the cystic fibrosis lung environment. One of the main blocks to research progressing in this field is that the in vivo lung infections are difficult to replicate in the lab. And even though we have a whole host of lab models to study this, they still are not completely replicating what you would see in a human cystic fibrosis lung. The go-to live animal model for these infections are mouse models. However, there's still a massive difference in the anatomy and physiology of a mouse lung when you compare it to a human lung, which means that those lung infections are still not truly what you would see in the clinic. We are developing an ex vivo pig lung model, as pig lungs are thought to be much closer to the human lung environment, but it also uses pig lungs as a waste product from the meat industry, which means that we also aim to reduce the need for live animal models in this research environment. So, looking at our ex vivo pig lung model in more depth, we receive the lungs from the abattoir on the day of slaughter, as you can see in this first image, and heat sterilise the surface of the tissues surrounding the bronchioles. We then extract these from the lungs, as you can see in the second image, and remove any excess tissue from the surface. We then passage it through a series of wash steps and cut it down to these small square pieces, as you can see in the third image, and UV sterilise them for 5 minutes. We then place them in a 24-well plate containing an artificial sputum media agarose pad. The artificial sputum media is a well-defined medium that has been developed to mimic the cystic fibrosis sputum. We then infect each lung tissue piece with Pseudomonas aeruginosa, in this case, using a needle and a single colony, and surround it with liquid artificial sputum media and incubate it at 37 degrees for the desired length of time. In doing this, we really want to work out whether this pig lung model is able to bridge the current gap that is seen between the lab models and what we are experiencing in the clinic. To do this, we first visualise the biofilm formed by Pseudomonas aeruginosa on our pig lung tissue, and then perform transcriptome analysis to look at the expression profile. So why are we interested in the biofilm in the first place? The Pseudomonas aeruginosa biofilm was long thought to follow a traditional lifestyle, forming a mushroom-like structured biofilm, which is a dense mass of cells grown together. As you can see in these first images of a Pseudomonas aeruginosa biofilm grown in artificial sputum media, they do seem to form this dense mushroom-style structured biofilm. However, when you look at patient biopsies, such as these second images taken from the bronchiole of a cystic fibrosis male with a chronic Pseudomonas aeruginosa infection, the biofilm looks a little different. This first image shows a gram stain infection, and these second two, hemotoxin and eosin. This is a histology stain used to stain both the tissue and the associated bacterial biofilm. As you can see, the Pseudomonas aeruginosa infection in these images seem like more of a honeycomb structure as opposed to the mushroom, with these small kind of gaps within the bacterial growth and the biofilm. So there's a difference here in the biofilm seen in the clinic and the lab, which could mean why we aren't seeing the same representation of the Pseudomonas aeruginosa in the lab that we are seeing in the cystic fibrosis lung. So we similarly performed the H&E staining on our pig lung model infected with Pseudomonas aeruginosa to see whether we were getting closer to that clinical biofilm. So the first image here shows uninfected bronchiolar tissue stained with H&E. As you can see, we have this clear structure of the epithelium tissue that doesn't appear to be infected with any contaminating bacteria, and the cartilage structure here. This second image shows a bronchiolar piece after infection with Pseudomonas aeruginosa for 7 days and then H&E stained. You can see this massive bacterial growth stained in purple on the surface of the tissue that is reminiscent of a clinical biofilm with this honeycomb-like structure, which is really exciting to see. We're much closer to those clinical biopsies than the current lab standards are. And if you look at the times 100 image, you can see the clear rod structures indicative of the Pseudomonas aeruginosa infection. And we also performed Alcium blue staining to stain the extracellular matrix of the biofilm to confirm that this was in fact the bacterial cells and the biofilm that we were staining with our H&E. So from this, we were able to say that we were accurately mimicking a biofilm structure more reminiscent of a patient biopsy than what the current lab standards are doing. So moving on from this, we then performed RNA sequencing to investigate the transcriptome of Pseudomonas aeruginosa grown in 2 independent pig lungs to compare to human infection to see whether we were able to bridge some of those gaps seen in the current lung lab models. So we grew the Pseudomonas for 48 hours, 7 days, and 24 hours as an earlier time point to determine the RNA expression. So we first looked at the pig lung environment, which was the lung tissue I initially mentioned, but we also extracted RNA from the artificial sputum media that surrounds this lung tissue to determine whether we had just a unified environment in the model system or whether in fact we had 2 clear distinct environments for the Pseudomonas growth. We also grew the Pseudomonas in vitro artificial sputum media as our kind of lab control condition, but also so that we could look at our results in context with other studies comparing lab environments to human infection. So this is our initial PCA plot showing the expression of the Pseudomonas in the 3 environments. This clear distinctly shows we've got 3 environments based on where the Pseudomonas was grown, which is really exciting. We've got a different environment in the model, but we're also seeing a different shift in time. From 24 hours growth in vitro, the blue circles at the bottom of the screen, you see that when you go on to 48 hours in vitro, they seem to move to the left in PC1 and up in PC2. In contrast, the 2 lung model environments surrounding ASM and the lung tissue itself, you see a shift from those circle dots at 24 hours to the right in PC1 and down in PC2 for 48 hours and the 7-day extended time point in the lungs. So this is really promising. We're not only getting 3 distinct environments, we're also seeing a difference in the shift over time, whether the Pseudomonas was grown in vitro or grown in the lung model environments. However, different doesn't necessarily mean that we're getting closer to human infection. It could have just meant that we were getting further away from it. So we also performed functional analysis to look at the KEGG pathways that are enriched by these differentially expressed genes. We compared the 2 lung environments to the in vitro ASM at both 24 hours and 48 hours. The bars to the left of the vertical lines show the differentially expressed genes that are underexpressed in the model environments compared to in vitro, and those to the right show the genes that were overexpressed in the lung model environments compared to in vitro. What we thought was particularly important to note was the enrichment of the phenosine biosynthesis pathway at both 24 hours and 48 hours in both of the lung model environments. The majority of these genes are involved in the piocyanin production pathway, which is an exotoxin associated with epithelium damage, as well as competition with other bacterial species in the cystic fibrosis lung. It is thought that this piocyanin is highly produced in the initial stages of infection. However, we see a reduction in its production as the Pseudomonas adapts to a chronic biofilm infection. So this is really promising that we're seeing an underexpression of these genes in our lung model compared to in vitro, suggesting that bacteria may be more readily or quicker adapting to this kind of biofilm lifestyle in our lung model compared to when it's grown in the lab. What's also interesting to note is the enrichment of the quorum sensing pathway. This is one aspect that is known that in human infection you see an upregulation of expression of these molecules, but it's actually thought that in the lab we are over-exaggerating this and we're actually seeing too heightened expression of quorum sensing molecules. So it was promising to see that a lot of these genes are being downregulated in our lung environment pathways. More specifically, it was mainly those in the LAS pathway. We then wanted to look at these genes in further detail, as this is one key aspect that's been shown in the transcriptome to differ between the lab and the human infection. So we compared it to a large study which looked at a host of lab environments and a host of human infection environments of Pseudomonas aeruginosa to compare their expression. They then pulled out 42 conserved quorum sensing genes, which have been shown to be present in all Pseudomonas aeruginosa isolates taken from the cystic fibrosis lung. As you can see, nearly all of these genes show a log-fold increase in expression in the lab environment compared to their human infection environments. I similarly pulled out this gene set from our data to compare our lab system to the pig lung model. As you can see, we have almost an identical profile, with the majority of these genes showing a log-fold increase in expression in the lab compared to our pig lung model. So this was really exciting. Not only is it different, but it also seems to be showing key markers of being closer to the human infection environment than the current lab model systems are. And this is part of some ongoing collaborative work with this lab. So in summary, we are able to replicate the key aspects of clinical infection that have not yet been possible in vitro. Not only is this a massive move forward in getting close to what we're seeing in the lab, in the clinic, but also means that the ex vivo pig lung model could be used as a potential diagnostic platform. Currently, when we're prescribing antibiotics, we may not be able to accurately determine whether an infection is going to be resistant or susceptible, because the way we're growing them in the lab is not the same as the way they are grown in the clinic. If they are grown more representative of a clinical infection in this lab model, we'll be able to more accurately prescribe an effective treatment plan. This will not only reduce the drain on resources and the financial burden, but could also improve the quality of life of people undergoing rigorous antibiotic treatments. As well as this, we may be able to use it to develop novel treatments, looking at the optimization of the current treatment plans together, or whether a whole new treatment to either act as an antibiotic or prevent infection occurring in the first place. As well as this, because we've been able to validate the key aspects of Pseudomonas biofilm infection, we can now use this model for future research with more hypothesis-driven questions, such as looking at mixed species biofilms and their spatial structure, as well as the drivers of chronic infection. I'd like to thank everybody involved in this work, particularly my primary supervisor, Dr Freya Harrison, and the rest of the lab, as well as Steve Quickly-Butchers in Leamington that provide us with the lungs, and everybody else. And thank you very much for giving me the opportunity to speak today.